Hi everyone, my name is Brenda Prieto, and over this past semester I worked on research having to do with enzymes. My research focused on the hydrolysis of peptide bonds using enzymes protease and proteinase in order to break down lidocaine using enzymatic activity under a green chemistry environment. In order to understand enzymes, I put a small picture showing how enzymes work. So a substrate will bind to the enzyme at the active site, and the enzyme will help the substrate become the products. For protease, the first enzyme, it catalyzes the hydrolysis of peptide bonds. It breaks down proteins into amino acids by breaking their peptide bond. This happens by having the nucleophilic attack using water attack the peptide bond. The other enzyme, proteinase, is a type of protease, but proteinase focuses on internal peptide bonds only, but it has the same function as a protease. When comparing proteases and proteinases, they both break down proteins into amino acids, as shown as the picture at the top. And on the left side, you see a protease breakdown, and on the right side, you see a proteinase breakdown. So a protease enzyme can break a peptide bond anywhere on the molecule, in the middle, on the sides, at any places, while the proteinase can only break down peptide bonds that are found internally inside the molecule. The hydrolysis of peptide bonds breaks down is when a peptide bond is broken down. So an amide group, which is shown by this first molecule, is broken down into amino groups. In order for this to happen, the amide group, which has the peptide bond, the enzymes attack that bond and break it. So when proteins are broken down, they are broken down into amino groups. And these enzymes are supposed to catalyze this reaction. So another substrate with an amide group should be catalyzed and broken down into amino groups as well. The substrate chosen for this experimentation was a lidocaine. Lidocaine is an amide that will be broken down into an amine group, is what we hope to see. The goal of this experiment is green chemistry. This is the approach of chemistry to maximize resources and minimize waste. So by using enzymes, we hope to use the ninth principle, which is known as catalysis. We hope to implement the protease's function on a selected substrate in order to determine if it is possible to break this substrate into its products while using aqueous conditions and these enzymatic activity. So the focus is catalysis because we're using enzymes. We are hoping to effectively break down the selected substrate, lidocaine, using both enzymes, protease and proteinase, under an aqueous environment while decreasing waste. Lidocaine, the substrate used for this experiment, is a local anesthetic used to hinder pain. It is commonly used to relieve pain under prescription. It has an amide group, which is shown by this nitrogen bounded to a hydrogen. And this is the bond between the nitrogen and the carbon that is attacked by the nucleophile through hydrolysis of the peptide bonds. The goal is to convert this amide group, lidocaine, into an amine group and a carboxylic group. This shows the mechanism of our substrate, which is lidocaine, and it is transformed into the two products, an amine group and a carboxylic group. Variables that affect enzymatic activity are temperature, concentration of substrate, concentration of enzyme, and the pH of the reaction. Variables used for this experiment: for temperature, we used 40 to 45 degrees Celsius. The concentration of the substrate varied, and the concentration of the enzyme varied as well, while the pH was maintained between 7 and 8. So for the proof of concept, the first experiment was performed by using water as a solvent for the reaction, lidocaine as a substrate, and the two enzymes. The volume for the lidocaine and the enzyme remained the same, and TLCs were performed in order to see separation from the lidocaine, the control, and the reactions that use the enzyme. These results suggested that the TLC number 3 showed the best separation with a more polar solvent, suggesting that a more polar solvent should be used for further analysis. The next experiment showed presence of products, and it used a lidocaine solution because I wanted to see whether I should keep lidocaine as a solid or use a solution. And in the RF values of this TLC suggested that the products were more polar than lidocaine, since lidocaine traveled further. This suggested that lidocaine should be kept as a solid for future work. For experiment 3 to 5, there was a change in substrate volume. TLCs were performed in order to see any differences or show presence of products. The TLC results from experiments 3 to 5 showed mostly that the products traveled further. For experiment 3, products showed that they traveled further for both enzymes. For experiment 4, protease showed that products traveled further, while proteinase were difficult to examine. The spots were messy and there was not enough information. For experiment 5, the products also traveled further. This suggests that lidocaine is more polar, suggesting that the products may not be the ones that we were looking for, suggesting that the amine group is not as polar as lidocaine. For experiment 6 through 8, there was a change in enzyme volume. The solvent used was still water, and the substrate volume used was 0.1. The enzyme value was changed, and there was 2 tests performed in order to determine the presence of any products. The first test was ferric chloride, and the second was an hydro test. So 3 tests are used in order to determine if there was any presence of product. For the ferric chloride, it is a colorimetric test. It shows the presence of carboxylic acid. A positive test is a color change to red, and a negative test is yellow or no color change. Bicarbonate test also shows the presence of carboxylic acid. A positive test would be the formation of bubbles, and a negative test would be no bubbles. A 9-hydrogen test would be a colorimetric test as well. It would show the presence of amines or amino acids. A positive test is a color change to purple, and a negative test is no color change. Any positive test would show the presence of some type of product. So results from experiments 6 through 8. For experiment 6, the ferric test showed that both enzymes were negative. For experiment 7, the 9-hydrogen test showed that protease enzyme was positive while proteinase was negative. For experiment A, both enzymes were positive. These experiments suggest that for ferric chloride, there shows no carboxylic acid, while non-hydro tests do show that there are some type of amines in our products. For experiment 9 through 12, we used a buffer. The buffer used was trisulfate, and the enzyme volumes were changed as well as the substrate volumes. For experiment 9, the results show for TLC there is not much shift in RF values, but it shows that products do travel slightly further. For a 9-hydrogen test, both came out positive, suggesting that there is an amine in the products. Our proteinase showed a darker purple. which may show that there are more product in that reaction, but it is unsure. For experiment 10, a 9-hydrogen test also came out positive. For experiment 11, the TLC had a shift, showing that products travel further, and for 9-hydrogen, both came out positive, which while having proteinase as a darker blue. For experiment 12, the 9-hydrogen test showed both positive, and a bicarbonate test showed both positive as well by having few bubbles. For experiment 13 and 14, collagen peptides was used as a substrate. This substrate was used in order to test the enzyme function and make sure they were working correctly. Collagen peptides was chosen because they can be broken down into amino acids. Amino acids can be determined using a 9-hydrogen test. For experiment 13, the TLC shows that the collagen peptides as a control travels further than any products. It shows that there are amino acids present using the 9-hydrogen test, and the bicarbonate test came out positive as well. For experiment 14, there is not much shift in the TLC. The 9-hydrogen test also shows that there is amino acids presence, which is expected since we are using collagen peptides as a substrate. For experiment 14 and 16, more buffer and a shorter reaction time was used in order to see any results that would show the presence of products. For these two experiments, 9-hydrogen test showed a positive test. It showed positive results in a dark orange-red color. A bicarbonate test also showed positive results due to formation of bubbles in all reactions. This is an example of a bicarbonate test done for experiment 16B. So as seen in the video, when adding bicarbonate to the sample, there is a formation of bubbles suggesting that there is a carboxylic acid in the products. To further analyze our results, IR spectrums were used. Lidocaine, the substrate, is shown. The spectrum on the left is received from the lab, and the spectrum on the right was found online under the NIST website. This shows that the spectrum from the lab may present lidocaine due to similar peaks at about 1500, 1750, and 3000. For the amino group product, spectrums were also taken from the lab and online. Spectrums from the lab show both enzymes protease and proteinase with similar peaks with the amino group found online. A summary of the results. The results from 9-hydrogen test showed the presence of an amine, which is a product that we were hoping to see, the 2,6-dimethyl aniline. Results for a bicarbonate test showed the presence of a carboxylic acid, which should be one of the products. Results for the ferric chloride test did not show the presence of a carboxylic acid, suggesting that there was no product present. And results for the TLC showed some presence of product, but not enough information from this type of chromatography. The IR spectrum showed the presence of the amine due to peaks at 1500 and 3200, which are both similar in the IR taken from the lab and IR taken online from the amine. Some limitations from this experiment are time, budget, equipment, substrates, and enzymes used. Future work would be to run samples through liquid chromatography, run samples through more instrumentation such as NMR in order to get the characteristics of the molecule, use other substrates and enzymes, and perform more reactions while changing variables that would affect the enzymatic activity. Some acknowledgments that I would like to give are to Dr. Kayasa, who helped me through this project, the OChem lab, which is where we worked, and some of my other lab partners, Nick Arnold and Natasha. That'll be all. Thank you all so much for listening.